Perfect. Okay, everybody, welcome to the May virtual roundtable for Hao-Fountain Syndrome. My name is Amber Freed. I am Executive Director, and we have Becky from the foundation as well as Bo. If other than Dr. Sumera, if we have anybody new on the call, feel free to introduce yourself. And I will add the link so that you can add yourself to our roster in the chat when I'm done speaking. There is a preprint of new research out, the mechanisms of USP7 MAGEL2 complex assembly and its mutational disruption in neurodevelopmental disease, with all of our trusted scientists as authors. I will send out that link in our wrap-up email. There are the typical funding opportunities. Last month, there was the funding opportunity through CIF, but you did need to be incorporated. Simons is refreshing some funding this month. I haven't had a chance to go through it, but I will include those links on the wrap-up email. We are underway in recruitment for our standard of care plan, which is very exciting to lay that foundational framework. Our disease concept study is nearly complete. We have a lot of matrix data that we are analyzing. And I'm going to, Bo, I'm putting you on notice that I'm putting you on the spot in a couple of minutes for any updates you have. We're also going along in developing a strategic research plan scientifically. We discussed this last time, but a standard of care is mostly a game plan for doctors as they diagnose patients of what to do. It's a basic framework as to how clinicians should be thinking about care for USP7 patients. There is a USP7 floxed heterozygous mouse available. The patient organization will pay the cost of the mouse and one-way airfare for the mouse to go wherever it is destined to be in this world. If you are interested, please contact me. We have lots of resources, as usual, that I'll include. What I more want to touch upon is that our symposium is coming up, and it is going to be the 9th and the 10th in Philadelphia. Luckily, that shouldn't be too far of travel for most East Coast people. If you haven't registered, please register. Thank you. I ordered Panera for the kids, and of course everything comes at once. We will be touching upon clinical resources. Combined Brain is going to be doing EEGs and collecting blood. And then, of course, we'll have so many scientific speakers. So if you haven't registered, now is the time. Start to expect an onslaught of emails in your inbox about this. Next month, we will be hosting Dr. Ingen and Dr. Meese from Yale to discuss a single patient case study and their frog model. And Bo, if you wanted to include any updates from your end, afterward, we can introduce Dr. Sumera joining us from Pakistan. and start the presentation. So I will stop sharing, and Bo, take it away with any updates you have.
Sure. I think you pretty much covered it, Amber. The one thing that we are also working on is a severity survey. We're working with Christian Schaff and his team in Heidelberg to try to get that up and running. Um, some of you may know that he worked on this with a student a few years ago, and that was sort of like the beginning, I think, when our group was a lot smaller, and it was based on families that were able to travel to Heidelberg and be part of the phenotyping study. And so, what we're trying to do now that we know we're in touch with a whole lot more people, and it doesn't require going to Heidelberg to be part of this severity survey. We're really just going to beef it up, basically, make it a lot better, make it a lot more robust. And he's got a student working for this summer that's going to work with us on that. So we're really excited about that. But otherwise, I think you covered everything, Amber. Thank you.
Sorry, I keep unmuting, but it's not. Awesome, Bo. And Dr. Sumera, we are so grateful to have you all of the way from Pakistan. Please feel free to introduce yourself, and maybe we can do a short round if of introductions, just so Dr. Samara understands. Like we meet monthly. These are all of the researchers that work on USP7. They're our trusted group, our little military helping, and hopefully you will meet them in person in October.
Thank you so much, Miss Amber. I'm going to start my presentation and I'm going to share it. So, let me just share my dry screen. Why I'm not sharing there? Just give me a minute. I'm trying to share the presentation. Right. I don't know why
I on the
the PowerPoint slides are not visible here.
on
I'm
the bottom toolbar,
trying to
you got it.
try to do this. Can you see my slides?
We can.
Hello?
We can
Okay,
you're doing
so
great.
today I'm going to talk about some work that I have done here in Karachi, Pakistan, which is basically about the identification of negative modulators of USP7 enzyme. I'm looking for, for the treatment against the cancer. So before I can actually talk about this research work, I would like to introduce myself. I have done my master in the field of biochemistry from University of Karachi. I have done my doctoral studies under the supervision of one of the most eminent scientists of Pakistan, Professor Dr. Muhammad Iqbal Chaudhry. And during my doctoral studies, I have worked on the identification of several molecules that have for the treatment of different pathological conditions, including tumor angiogenesis, oxidative stress, and CNS myelin pathologies. I have been awarded with 6 month split PhD program in national in some in University Paris Sud France, and then I have proceeded for my postdoctoral training under Milan and Dr. Virginie in France, and where I have learned various aspects of molecular and structural biology. And there I have also learned various techniques of biomolecular NMR spectroscopy. Uh, uh, what happened? Something wrong. Okay. Currently, I am serving as assistant professor at Dr. Panjwani Center for Molecular Medicine and Drug Research. This is one of the institutes located in the University of Karachi. This institute basically has work both in the field of chemistry and biology, and my research interest basically includes the drug discovery using molecular structured biology technique. I also have expertise in using in utilizing biochemical and biophysical techniques to identify the compound that can interact with the proteins. Basically I try to identify the compounds that possess the ability to interfere with the biological function of those enzymes that have role in various diseases. So if we talk about my expertise, I have been trained in I have training to produce the recombinant proteins using the prokaryotic system in E. coli. I have the expertise to transform, to express, to purify using various chromatographic techniques, including affinity, size exclusion, ion exchange. And I also have the training to perform the ligand based and target based NMR spectroscopy. And today I'm going to show some of my work which is related to recombinant protein production of USP7, and then I will talk about the utilization of STD NMR, how we can use that technique to identify different compounds that bind to the ubiquitinase. I also have training to optimize or establish new enzyme inhibition assay using UV visible spectroscopic technique and many others. The targets on which I am currently working include thymidine phosphorylase, some of subunit of transcription factor 2H, monoamine oxidases, D-ubiquitinases, ribosyl phosphorylases, and various drug targets of H. pylori and Salmonella typhi. Various students have been completed their MPhil studies, and their some of them are also doing some doctoral studies under my supervision. I'm also supervising various master's student and BS thesis students. Apart from research, I also have a role in teaching various aspects of subjects like physical biochemistry, molecular genetics, biotechnology, some techniques of the molecular medicine, and also protein structure and function. So today, let's start with the deubiquitinases. Why I am, how I'm using this as target. As you guys have a very different role of the USP7 enzyme, I am using this as a drug target for the development of anti-cancer uh, drugs and molecules. So let me just give you a very brief overview that D-ubiquitinases are the enzymes that are responsible to remove the ubiquitin tag from the protein that have been destined to be degraded by the proteasomal system. And these have a very important role in the protein homeostasis, in DNA damage repair, in cell signaling. And we all may know that there are two major classes of the ubiquitinases, including the cysteine proteases and the metalloproteases. And among the cysteine proteases, we have this ubiquitin specific protease that has 58 members included in it. And I'm sorry, I have some, I think there is some issue with the PowerPoint app. As I'm talking USP7 is with specific respect to cancer. So here I have shown different aspects of tumor biology that are being influenced by the D-ubiquitinases. These enzymes basically responsible to allow the cancerous cells to resist the cell death. They allow them to proliferate. They also cause them to evade or to avoid the growth suppressor. They cause the genomic instability. They are responsible to cause the tumor promoting inflammation. They also cause the dysregulating cellular energetics. So, uh, here the specific pathological role of USP7 with reference to cancer, that one of the protein which is known as MGM2, which is once ubiquitinated, this cannot work on the P53, which is one of the most known tumor suppressor protein. When this enzyme is functional, it causes the deubiquitination of this protein, and once this protein is deubiquitinated, it causes the ubiquitination of P53. So the idea is that if we somehow interfere with the function of this enzyme, the ubiquitinase, then it will remain in the ubiquitinated state, and we can restore the cellular concentration of P53, which once present in its in good quantity, it can promote tumor suppression. So, talking about the structural feature of this enzyme, I suppose and I believe that you guys may know that it is a big protein that has around 110 kilodalton. It has a catalytic domain. It has a TRF binding domain and ubiquitin binding like domains. And in this, we have the catalytic domain is arranged like it has finger at one side, thumb at one side, and palm another side. So these are the various identified inhibitors against this enzyme. However, none of them have been proceeded for the clinical trial or clinical use, but they are, some of them are associated with severe side effects. So there is still need exists to identify new molecules and compounds that can interfere with the function of this enzyme. So what was our idea is, we actually transform the plasmid containing the gene of USP7 into bacteria. We express them in E. coli and then we purify that. Here, I would like to tell you that we initially tried to express the whole enzyme, but we couldn't succeed in that. So later on, we just purified the catalytic domain of that enzyme. And after having protein in hand, we screen the FDA approved drugs that we obtain from the drug bank of our institute. And here we actually use this approach, which is drug repurposing. Repurposing is one of is I believe that you guys may know about drug repurposing approach. Is one of the mostly technique that approach that actually decrease the time associated with drug development. The compound that we obtained, we screen them via STD NMR, and then we studied the compounds by thermal shift assay, and then we perform the molecular docking to get the structural insight into protein ligand interaction. Ligand here is the same as the drugs that we found active, or that we found binding to the protein. Then we perform the SCD titration studies to determine the affinity between the ligand and protein, and then we perform certain cytotoxic studies on the normal human fibroblast cells and colorectal cancer cell line, and some gene expression studies. So here is the brief layout that has the information about the plasmid construct. This plasmid we actually obtained from one of our collaborator, and this has the gene of catalytic domain of USP7. We transform it into E. coli BL21DE3 P. lyse cells. Then we perform the colony PCR on the colonies that we obtain, and after having a positive outcome from them, we proceeded for the small scale protein production. After having confirmed that we have protein producing from the system, we produce large scale production of this protein, and we purify this via affinity chromatography followed by the single size exclusion chromatography. We analyze the fraction using SDS page, and then we run the proton NMR to see the protein that we obtain has retained its 3D fold or not. After that, we took that protein for the biophysical method, as I mentioned in my previous slides, that I have used NMR as a major tool to identify the compound that showed interaction with the protein. So, for those who are new, for those who for those uh, this terminology, for those who are new, I can tell you that this is actually a saturation transfer difference nuclear magnetic resonance technique, where what we do is that we selectively saturate our protein into the region where there is no existence of ligand resonances, and then that saturation transfer to the entire surface of protein via spin diffusion. And if your ligand or compound or your drug which you are trying to study is present in close proximity to your protein, it will also receive that saturation, and you will see that saturation into your difference spectra. Here you basically record two types of spectra, which is in first experiment you give the saturation in a region where you have the protein resonances, while another experiment you give saturation to the region where there is no such resonances from either your protein or your compound, and then you subtract these two experiments from each other, and you got a difference spectrum where there where you will find only the resonances from the compound that have interacted with the protein. So I will show you how we interpret in my upcoming slides. And after having studied them via STD NMR, we proceed them for the thermal shift assay, where basically we expose our protein to higher temperature from 25 to 95 degrees centigrade, and when the hydrophobic residues of the protein become exposed, there is one dye present which is cypro-orange that bind to these hydrophobic region, and it produce the fluorescence, and that actually help us to identify the melting temperature for a particular protein, where it will be half folded and half unfolded. This experiment basically tells you about the thermal stability of protein in the presence and absence of protein. Here, I would also like to tell you that I did not perform the enzyme inhibition assay, as the assay for the enzyme inhibition, the substrate was very expensive, so I couldn't perform that. So I use these biophysical methods to screen or to identify the compound that bind or negatively shift the stability of the protein. So from I have performed the nuclear magnetic based screening of 172 drugs. These drugs are being present in the group in the form of mixture, and each mixture it contain 4 to 5 drug in it. So let me try to start the result and discussion. This is basically a proton NMR analysis of any mixture. Here I am showing you the analysis for mixture 2. The A actually represent the proton NMR for all the compounds which are present in this mixture, while B, C, D, and E are the proton NMR of the individual compounds or drugs which are present in this mixture. After confirming that your mixture contain all these compounds, and we can actually locate, or we can actually trace that which resonance is coming from which compound, we proceeded that mixture for the STD NMR. The idea of using mixture is that in limited time you can screen multiple compounds. You can run the STD NMR on these compound individually as well, but in that case the experimental time will be too much. So once you should, if you are using the mixture analysis approach, you will be rapidly screen the compounds, and you will be able to identify that which mixture contain the compounds that interacted with the protein. So after having their proton NMR analysis, we perform the STD NMR on this mixture. And let me explain first that what is what. This A is the proton NMR, same as that of this one. The red actually represent the difference spectra recorded in the absence of protein, and this green represent the difference spectra recorded in the presence of protein. The idea of having a difference NMR recorded in the absence of protein to nullify the false positive result. Ideally, if your compound is interacting with your protein, it will show you resonances in the different spectra recorded in the presence of proteins. Here, we cannot see any resonances, indicating that this mixture does not contain any compound that showed interaction with the protein. So here, the the STD NMR analysis for mixture 6. Again, this is difference spectra recorded in the absence of protein, and this is in the presence of protein. And here you can see that some of the resonances which are present here also present in this difference spectra, indicating that this mixture might contain some compound that interacted with the protein. So after knowing that mixture contained the compound that showed interaction, we run the STD-NMR on all the compounds which are present on that mixture. And here we found that a compound that with the code of DB439, which is basically a code of diclofenic sodium, a drug that I'm going to show in my next slides. It's the one that interacted with the protein. So we have discovered 30 compound from mixture number these that showed interaction with the USP7 catalytic domain in the STD NMR experiment. And when these 30 compounds evaluated individually for the STD NMR, we found only 11 compounds that retain their ability to bind with the USP7 catalytic domain. So after that, I'm going to talk about the group epitope mapping, or basically a group epitope mapping is kind of study where you basically can map the orientation of a compound on the protein site. So this compound, which is DB439 that I showed you in your STD-NMR spectra, it is a bromophenolic sodium, an anti-inflammatory and analgesic drug. And if you look into the structure of this compound, you will see that there are various proton present that can be studied very easily using the NMR. And here you may be seeing different percentages. Here I have given 100% saturation received by these two proton. I have given 100% value to this proton because while interpreting the STD data, the resonances that show the highest integral values, they are usually designed or they are usually assigned to receive 100% saturation from protein. Percent saturation of other proton are calculated relative to this. So here we can say that these two proton, they are present in the near proximity of the protein, followed by proton present at 3' or 5'. And this indicates that this moiety, which contains the bromine substitution on it, is present in close proximity to protein, followed by this moiety. As you can see that the proton present in this ring, they have given or they have shown 73 to 78% of relative saturation. I would like to tell you again that this is the proton NMR of this compound, and this is the STD difference spectra of this protein. Then another compound, which is diclofenic sodium, where the proton present at 3 triple prime and 5 triple prime, they have been assigned to receive 100% saturation from the protein, followed by a proton 3 double prime that receive 87% of the saturation. Here we can also propose that this ring is present in close proximity to protein in comparison to this one. So similar is the case for another drug, the ketoprofen, that has been used as anti-inflammatory drug. And then we have another drug that is labetalol, ofendrine, oxybutynin, ketotifen, ticlofidine, and lamotrigine pantoprazole, sodium, escitalopram, and after mapping the orientation of these compound on protein, we next took these compounds for the thermal shift assay. That you can see that here I have shown you the thermal shift profile of USP7's catalytic domain in the absence of protein, and we can see that when there is no compound present, it gives you a TM of 43 or 45 degrees centigrade, while in the presence of compound, the TM may lower or may increase. If some compound is increasing the TM of your protein, it means that that compound is actually stabilizing the 3D fold of that protein. And those compounds which are decreasing the TM, they are supposed to destabilize the 3D fold of that particular protein. So here is the bar diagram of those compound that I have identified using the STD NMR technique, and you can see that most of the compound, they are responsible to decrease the TM of the protein catalytic domain, which means that these compounds may have role in interfering, or they may cause the destabilization of USP7 protein. And we may propose that when studying them via enzyme inhibition assay, they might interfere with the biological function of this enzyme. After studying their profile on the thermal stability, we next studied these compounds via computational. This part is being done by one of my collaborator, Dr. Humaira Zafar. And here there is catalytic tride present, and we found that most of our compound, they are interacting in the thumb palm cleft of the catalytic domain of USP7 protein. We next proceeded these compound for the cell culture, and where we performed the cytotoxicity assay and performed certain gene expression analysis. These compound, these were found to be safe for the BJ cell line. BJ cell line is basically a human fibroblast cell line, and some of the compound like compound 6, 7, 10, 11, they showed cytotoxic behavior towards the colorectal cancer cell line, that is HCT116. We then performed the effect of these compound on the mRNA expression of USP7, MGM2, P53, and c-MYC. These are the proto-oncogenic genes. So we actually believe, or we actually suspect that these compound might decrease the mRNA expression of these genes, and increase the mRNA expression of this P53 gene. And luckily, what we found that the compound 6, it caused a decrease in the expression of USP7, MGM2, and c-MYC, and it also increased the expression of P53. However, it is non-significant. Similarly, for compound 7, it also caused a decrease in the expression of these proto-oncogenes, and also it increased the gene expression of P53. However, this is lesser than that of the standard compound, and it is also non-significant when compared with the normal cells. And similarly for the compound and 10 and 11, that these compounds were basically causing a decrease in the mRNA expression of proto-oncogene. However, their effect on the p53 is not very significant. So I can conclude that this study has identified several drugs that found that interacted with the protein during the STD NMR, and they also cause reduction, or they also cause the destabilization of protein fold as identified from the TM values. So these molecules need to be evaluated via in vitro enzyme inhibition assay, which I told you I couldn't do them because of the expensive substrate, and they need to be evaluated at in vivo level. From this study, I have published two research articles that have been published in Helion in RSC Advances. And I would like to acknowledge some people who actually work with me during this research work. Miss Seema Zadi is my student who actually performed these experiments with me. Dr. Humaira Zafar, they actually performed the computational part. Dr. Atiatul Wahab, I have performed these experiments in her lab, and Professor Dr. Muhammad Iqbal Chaudhary, who is actually my supervisor, from whom I learned so many things. So with this, I think I ended from my side. So I would like to stop sharing, and I would like to take questions from you. So now my presentation is done. I can take a question from you guys. I hope that it is a different theme of work. I hope that some part of it is easy to understand for you guys.
Hello. Can I ask a question? Can you hear me?
Yeah. I can hear you.
Well, this is very good work, and I congratulate you. It's a lot of work.
Thank you
We
so much.
were one of the first company to identify the inhibitor for USP7. This is Tosif Butt calling from Malvern, Pennsylvania.
Okay.
And we published some of the work on USP7. We'll be happy to help you with the substrates and other things you need. It seems to me you were unable to test the compound for enzymatic activity. So if you need any help with the substrate or compound, we are happy to help you. Let's see if I can leave my name and email address in the chat so you can contact us.
Yeah, it will be of great help, because I have obtained substrate 1 to check that whether the catalytic domain that I'm purifying is active or not. I could get its activity, but unfortunately it was very expensive. I could not perform the enzyme inhibition assay on that. So therefore I couldn't do that. So
No,
if you can.
I understand that, but I visited your center a long time ago, so
When?
I'm a little bit familiar. But you can communicate with us, and you can send us an email. We'll be happy to help you. My email address is
That's
in the chat. Okay, good work.
okay. Thank you so much. I will contact you definitely, because right now I'm also working one of another enzyme that is D-ubiquitinase 2, which also has a role in some of the pathologies. So I will definitely contact you. When did you visit our institute?
120 years ago.
120 years ago, long time
Well,
ago.
when it was just being formed.
In in 1960s, 70s.
No, I think it must have been 90s.
You're talking about specifically the Dr. Panchwani Center. So it was
Right,
in 2000. Yeah, it was
right,
in
right.
2004. So maybe. So when are you visiting us again?
No, no. I'll be happy to, but I'm in Philadelphia and I work in Progenera.
Okay.
We
Okay.
published
Thank you so.
a lot of work on USP7, so you can look into it with Dr. Wayne Hancock and other people at University of Pennsylvania. But
Sure, I will.
yeah, please take a look. Yeah, thanks.
I will.
I think our friend Benedict Kessler we are we talk once in a while.
Yes, good to see you, to see alive and kicking. That's great. We have also worked together in the past, actually on the first series of USP7 small molecule USP7 inhibitors. See how the field has grown after that. To see this
Yeah,
is quite good to
it's
see.
very
Actually.
gratifying.
Yeah. So very nice presentation. If I may allow also to ask one or two questions.
Yeah, please, please.
It's
Thank you
great
so much.
to look for small molecules and small molecule modulators, but compounds like bronophenac and others, they may also have other targets. The likelihood that you may also target, in addition to USP7, something else is,
Yeah.
at least at that stage, is probably quite high. Can you, maybe I didn't catch it, but can you tell us what was the criteria to select these compounds that you used for your screen? What was your criteria?
Okay, the reason I selected these drugs is number one. These are drugs, and I wanted I wanted to utilize them for the drug repurposing approach. Another thing, I'm working on the STD NMR, and that for that I need to have a drug that is soluble in water, because if you might know that the STD NMR has a limitation that it only work or it only useful for the compounds that are water soluble. No specific reason of using these screening these drugs. I have mentioned you that I have screened 172 drugs, and among them I found these drugs that interacted with this protein during the STD NMR. So there is no very specific reason of using these drugs. We have around 500 to 700 drugs stored in our drug bank of the institute. So what we do is to screen them and find out that which drug is able to interact with the protein.
Yeah. Thank you very much. Thank you.
Welcome. Thank you.
I have a question as well, if you don't mind.
No,
Can you
I
hear
will
me
not
okay?
mind. Please ask. Yeah, I
Okay,
will
yeah.
be okay. Please ask.
Thanks for the presentation. It was really great. I just wanted to ask about the type of protein that you're using. So you're using only the catalytic domain of USP7. Do you plan to repeat some of these experiments with either the full length or just the C-terminal tail? Because it does impact the activity of the protein quite a bit.
So I told you that I tried to clone the full length of this enzyme, but we were not able to obtain the protein. We could the transformation was successful, but the protein production was not successful. So therefore, I limited myself with the catalytic domain. So the catalytic domain, the plasmid for the catalytic domain, I obtained from one of the collaborator that we have that I mentioned in my research article. I don't remember the name of that lab. It is Dr. Turnbull who actually shared his plasmid with me. I usually use the services of GeneScript to synthesize the construct, the plasmid construct. I tried, but I couldn't succeed.
Sumaira, it's difficult to express full-length protein in E. coli. We have
Yeah,
had a lot of experience. So
yeah,
it's normally expressed in mammalian cell or insect cells.
yeah,
Um,
yeah,
then again, I'll find out. I think we have some protein, but if you wanted to, I would express it in insect cells. It's quite okay. Don't try to spend time making full-length
yeah,
protein in insect cells. I
yeah.
mean, sorry,
You
E.
are,
coli.
you are, yeah, you are very right. Actually, currently I don't have the expertise of using the insect cell or the mammalian expression system. I would like to learn that in my future research. Currently,
Yeah.
I just have the experience of with the prokaryotic system only. Therefore, I just tried. I just tried. I wanted to look
We
whether it
can we can
express or
give
the,
you active protein. We
you
can
can
give
give
you
me.
full-length active protein.
Oh, that's great. That will be so nice of you. I will definitely contact you, Dr. Tausi. Thank you so much.
You're welcome.
Samira, I have a question.
So, yeah,
Uh,
please.
I heard you mention lamotrigine, and I know it seemed like you were saying it was one of the compounds that physically bound to USP7's catalytic domain. I was just wondering if you made any other findings specific to lamotrigine. I'm just curious.
No, no. I did not do not perform any other further studies. I will, but currently I just have these studies that I have shown to you.
Okay, thanks.
Thank you so much.
All right. Well, is that all of the questions? Bo, I only see you. So,
I think that is all the questions. Yeah.
okay. Well, I will send the recording out. And if anybody has follow-up questions, I will include all contact information and everybody go kill Tuesday. Bye.
Thank
Thank
you.
you. Yeah.
Bye.
Bye.